Today is going to be talking about DNA replication. And up until this point, what we've really done is look at the cell cycle, look at mitosis, and describe how these things work together. And then we started to really dive in and look at what is that structure of DNA, what's the history of it, what does it look like, how is it built. And so now what we're going to do is to move forward and look not only at what we know about the structure of DNA, but now we're going to be able to discuss and really come to terms with how DNA replicates or how it copies itself. And so our topic is DNA replication, and our essential question that you should be able to answer at the end of this discussion in a lot of detail is, how does DNA replicate? So first of all, let's just start with DNA replication. Remember that DNA replicates during the S phase of the cell cycle. And so after G1, the cell enters into the S phase. At that point, all of the DNA is copied and gets ready for the cell to then go into the M phase or into the mitotic phase. And one of the fun facts that I like to share at this point is that in your lifetime, you will produce enough DNA will be replicated, in other words, that you could fill the entire space for one entire light year. That's a massive amount of DNA that is able to replicate and do so very efficiently, quickly, accurately. And so, how does DNA replicate? Well, like I always do, let's start with a brief history of DNA replication and the models that came about when we were trying to figure out how this works. There were three different alternate models that came out for DNA replication. And of course, these models are going to be built around the work of Watson and Crick, because they had just published their paper in 1953. So we were sure of what the structure of DNA was. Now we were looking at how it replicates itself. There were three models that were proposed, and the three models are the conservative model, the semi-conservative model, and the dispersive model. So what did each of these models propose was the way that DNA replicated. In the conservative model, the parental strands direct synthesis of an entirely new double-stranded molecule. And so the parental strands are fully conserved or saved. And if you look at the diagram here, you can see that the gray parental strands are completely intact as we get down to the two daughter molecules. But the blue would represent two completely new strands of DNA that would make up the second daughter molecule. The other method that was proposed for DNA replication was referred to as the semi-conservative model. And in this model, the two strands are going to separate and each makes a copy of themselves. And so after one round of replication with the two daughter molecules, each one of those molecules has one parental strand and one new strand. This was in fact the model that Watson and Crick proposed after they got the structure of DNA correct. Finally, the last of the models is what is referred to as the dispersive model. And in this model, the material in the two parental strands is dispersed randomly between two daughter molecules. And so after one round of replication, the daughter molecule contains a random mix of parental and new DNA. Which model then is correct? Well, in 1954, almost exactly 1 year after Watson and Crick published their paper, Michelson and Stahl performed an experiment using bacteria. So the process that they used was that the bacteria was cultured with a heavy isotope, nitrogen-15, so extremely heavy nitrogen. And we'll remember from our study of macromolecules, nitrogen is a key component of all nucleic acids. And so because it is part of the nucleic acid, that nitrogen-15 became part of the cells that were cultured on that bacteria. Once the bacteria had been cultured several times, they were transferred to another medium that was composed of nitrogen-14, which was a light isotope of nitrogen. And after every round that the bacteria replicated or went through a life cycle, their DNA was centrifuged to see how dense it was. So remember, the DNA that's made from N15, that really high, heavy isotope of nitrogen, is going to be very, very dense or heavy, versus the lighter isotope is going to, nucleic acids are actually going to be lighter. And so by centrifuging it, they were actually able to see how much of that N15 was still in the genetic material of the bacteria. And so by analyzing the samples of DNA after each generation, it was found that the parental strands followed the semi-conservative model. And if we look here at the data table, you can see that as the generations progressed, there was more and more of that N14 present. However, that N15 never completely went away. And so Michelson and Stahl, through very precise experimentation, were able to show that semi-conservative model to be correct. And if we really look at what that means, you can see here in the diagram, as each of those generations go on, the dark red represents the initial DNA strand that was made from the N15, and you can see how even down to generation 4, so that DNA is replicated 4 times at this point, that N15 is still part of the DNA. And that really brings us to how DNA replicates. So first of all, it's important to remember that DNA replication begins at sites called the origins of replication. In eukaryotic cells, these origins of replication are going to be in hundreds of places throughout the cell, or throughout the DNA molecule, excuse me. And so DNA in eukaryotic cells does not replicate tip to tail. It replicates in multiple places at once. And so those are all referred to as origins of replication. At the different origins of replications, various proteins are going to attach to that origin, and they're going to open the DNA to form a replication fork. And in this diagram, your replication fork is shown here, where it actually opens up and is no longer that double helix form. So just like we know proteins are one of the most important molecules in the human body, you're going to see now we have a series of proteins in the form of enzymes and just structural proteins that actually allow for DNA replication to occur. The first enzyme that you need to know is called helicase, and helicase's job is to unwind the DNA strands starting at each replication fork. And so remember, it's going to break those hydrogen bonds, but hydrogen bonds are like magnets, right? Like they want to snap back into place. And so if the helicase goes through, it breaks the hydrogen bonds. But if there wasn't something else there, the DNA would reclose on itself. And so what comes right after the helicase are single-stranded or single-strand binding proteins, SSBPs, and they're going to bind to the DNA to keep it open. They're like door stops. Ahead of the helicase then is going to be another enzyme called topoisomerase, and this is going to help to prevent strain on the DNA molecule from stretching it open. So remember, it's tightly coiled. It wants to stay tightly coiled because that's how we package it so well in our cells, 6 feet of DNA in each of our cells. And so if we didn't have topoisomerase, that strain would get too great and the DNA molecule would actually break. And remember, from DNA extraction, we know that it's a pretty fragile molecule anyway. And so topoisomerase takes its place. And so if we look at what this looks like, here's our DNA strand model. Let's go ahead. There's our helicase. After that comes in our SSBPs. And then finally, our topoisomerase comes into place. So now we're sitting, our DNA has been opened. It's ready to start replicating. What's the next step? So an enzyme named primase, or RNA primase, initiates replication by adding short segments of RNA called primers to the parental DNA strand. And so we use RNA because the enzymes that actually make DNA can only attach new DNA nucleotides to existing strands of nucleotides. So we can't actually start with DNA, we have to start with RNA. Primers then become the foundation for DNA synthesis because they are what allows it to occur. So let's look. Here's our picture where we left off. Topo is in place. Helicase is ready to keep going. Our SSBPs are in place. So now we need to bring in primase, and there's our RNA primer left behind. Once our primer is in place, now this is where things on the two strands get a little bit different, because remember, our strands are anti-parallel to each other, meaning one runs 5' to 3', the other runs 3' to 5'. And so because they're sitting anti-parallel to each other, when it replicates, we also have to take that anti-parallelism into consideration. So we have several DNA polymerase molecules, and you will be learning about two of them. Specifically, the first one is going to be DNA polymerase 3, or DNAP3, which attaches to each primer on the parental strand. And DNAP3 is always going to move in a 3 to 5 direction, and as it moves, it builds the new strand in a 5 to 3 direction, and it is limited by that directionality. DNAP3 then is on both strands. The strand where DNAP3 follows helicase is known as the leading strand, and it only requires one primer. The DNAP3 on the other parental strand that's acting as a template is going to move away from helicase, and that becomes known as our lagging strand, and it's going to require many primers. So again, here we are, we're all set up. You can see you have everything in place. We have the leading strand, which has one RNA primer on it. We have the lagging strand, which has 2 DNA primers on it. So DNA polymerase 3, remember, moves 3 to 5 following the helicase. So we can see there's my enzyme polymerase. There's the directionality that it's going to be moving. On my lagging strand, there's my polymerase, but remember, it's going to be moving away from helicase because it also has to travel in a 3 to 5 direction. So if we look at what happens when these move, we'll be able to see what happens and how that all works together. So step 5 then, the leading strand is going to be synthesized in one continuous segment. And so that DNA polymerase just goes through and it just chunks those nucleotides right in place, right along the train track there. But since the lagging strand moves away from the replication fork, it actually ends up being synthesized in chunks. And those chunks are referred to as Okazaki fragments. And it's called Okazaki because of the last name of the wife and husband scientist team that discovered this process. And so again, here's our picture. Let's watch what happens when things actually move. There goes my polymerase 3 on my leading strand. And then if we watch the lagging strand, you can see how it kind of works backwards. It makes those chunks. So right there is what we refer to as an Okazaki fragment. After DNAP3 then has formed an Okazaki fragment, now we get into our second molecule of DNA polymerase that you need to be aware of, and that's DNAP1, or DNA polymerase 1. And its job is to replace all of the RNA nucleotides with DNA nucleotides. Once DNAP has gone through and removed all of the RNA, DNA ligase is going to come through, and it acts as a glue, and it joins all of the Okazaki fragments together, forming continuous DNA strand. So let's finish up our DNA replication here on our model. There's my DNAP1. It got rid of my RNA on my leading strand. DNAP1 getting rid of my RNA on my lagging strand. And then ligase comes through and connects the strands. So let's watch this video together and really see how all of this works as one continuous process. So there we have DNA replication in the whole, but there are a few things that we need to address, the first of which being the problems at the 5' end. So since DNA polymerase 3 can only add nucleotides to a 3' end, there's no way to finish the replication on the 5' end of the lagging strand. And so what would happen if this wasn't addressed was that over many replications, the DNA would become shorter and shorter and not function. And so genes need to be protected against this. And the protection that we have in our bodies are called telomeres. And a telomere is a repeating unit of short nucleotide sequences that don't actually code for genes. They just cap the DNA and they help to postpone or prevent that erosion of important material. And again, we have another enzyme here, telomerase, which actually adds the telomeres to DNA. Proofreading and repair is going to occur in two ways. One, it happens while the DNA molecules are being built, and then it happens after they've been built. And so as DNA polymerase adds nucleotides to the new DNA strands, it proofreads the bases and make sure that it's adding them or has added them correctly. If errors still occur, it will quickly do what we call a mismatch repair, where enzymes will remove and replace the incorrectly paired nucleotide. And this can either be just a generalized exonuclease or it can be DNA polymerase 1. If whole segments of DNA are damaged, then a nuclease comes in and removes an entire segment of nucleotides, and then DNA polymerase and ligase go back through and clean up by replacing the nucleotides and gluing the new segments together. So on this portion of our quick review, what I would like you to do is to read the question, pause the video, answer it, and then check your answer. So the first question is, if a parental DNA strand reads 5' ACGTAC 3', what would the new synthesized complementary strand read? And your second question is to summarize the role of helicase and topoisomerase and DNA polymerase 3. Pause the video, answer the questions, then when you resume the video, check your responses. What direction does DNA polymerase 3 move along the parental strand? Pause the video, answer the question, then check your answer by resuming the video. Why do Okazaki fragments occur on the lagging strand? Pause the video, answer the question, then check your answer by resuming the video. All right, we have gotten to the end of our discussion on DNA replication. There's a lot of information that has just been given to you. So please take the time and complete a summary of your notes by answering your essential question. And remember, your essential question is to describe the process of DNA replication in great detail. If you have any questions,